Genes of you and me, genes of you, and you're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same.
Hello, and welcome to the 21st episode of Discovering New Advances, or DNA, a podcast that keeps you up to date about the scientific world in an easy-to-understand language, so whether you're a student in middle school or a grandparent, you'll easily be able to follow along. I'm Kieran. On this episode, I will give an introduction lesson about gel electrophoresis. For more information, please visit dnapodcast.com or email in at info@dnapodcast.com. Gel electrophoresis may sound like this big fancy phrase, but the process is relatively simple. So simple that I used gel electrophoresis twice back in high school, in AP Bio, and in a biotechnology course, which was academic level. So let's find out how this simple process works and what it's used for. Gel electrophoresis separates macromolecules such as proteins, RNA, and most commonly DNA. It takes one of these macromolecules already broken down and separates the pieces by their charge and size. Just as the name suggests, there is a gel, and this gel has wells where the pieces of macromolecule are placed. The wells are on one side of the gel. There is a current that goes through the gel with a positive charge on the opposite side of the well and the negative side on the well side. DNA has a slightly negative charge due to the sugar phosphate backbone, which is the sides of the DNA. Therefore, the negative force pushes the molecules and the positive force pulls the molecules towards it, so therefore the molecules of DNA are going through the gel. The smaller molecules move faster than the larger molecules, as the smaller molecules have less resistance against the gel. It is possible for these fragments to move through the gel because of the small holes in the gel. It's kind of like Jell-O, which I'll be referring to throughout the episode. When the gel has been run, the smaller pieces of DNA are further than the larger pieces, which can be sorted in this manner. In order to see the macromolecules in this gel once it's been run, the gel is stained so the molecules are visible to the naked eye as bands. There are different stains used depending on the molecule. A gel with DNA run on it has stained with ethidium bromide. When ethidium bromide interacts with DNA, the DNA is fluorescent under ultraviolet light. Usually, pictures would be taken of the gel with the stained bands and marked with a scale of base pairs to identify how long the DNA pieces are. This gel is made of purified agarose. To make the gel, the liquid purified agarose is heated, then poured into the mold. It then solidifies for use, just like Jell-O. You heat it up, you pour it in a mold, and then it solidifies. When running the gel is placed in a pH buffer to control the pH changes due to the electric field. Electrophoresis and gel electrophoresis refers to the electromotive force used to push and pull the molecules through the gel. We have touched upon it a tad, but there are three main applications of gel electrophoresis. The estimation of the size of DNA can be conducted after a restriction enzyme has digested the DNA to analyze its products. The other application of gel electrophoresis is to analyze PCR products. There will be an episode in the future about PCR, but a quick explanation. PCR stands for polymerase chain reaction, and it amplifies a sample of DNA, producing thousands to millions of copies of that one sample of DNA. Gel electrophoresis can analyze these products such as molecular genetic diagnosis or genetic fingerprinting. The third application is separating DNA before a southern transfer, another term you'll learn more about in the future. But basically, the southern transfer takes the DNA fragments from the gel and filters them through a membrane, then uses probe hybridization to identify certain DNA sequences, which kind of is markers and it highlights certain parts. So let's review what we've learned. Gel electrophoresis is a method of separating macromolecules such as DNA. The system works by having the DNA in the wells on one side of the gel. An electric current runs through the gel, moving the molecules through the gel. The smaller pieces move faster than the larger pieces. Once the gel has been run, it is stained, and the molecules can be seen as bands in the gel. A picture is then taken of the gel, and a scale is drawn to, with the number of base pairs as its key. Size and fragments of the DNA can then be estimated. I hope you found this episode educational and informative on how gel electrophoresis works. Again, it's not that difficult. You put the DNA in the wells, you turn it on, it moves it, and then you can see the different fragments and size of them and charges. So if you want to learn more about genetics, you can go to dnapodcast.com for further episodes along with the audio formats and a visual format of transcript. If you want to contact me, you can go to the website and click the contact form, or you can email me directly at info@dnapodcast.com. If you're a social media junkie like I am, you can follow me on Facebook and Twitter, both at /DNA Podcast. So to keep up with all the new releases, if you don't want to figure out when I'm releasing, but it is the first of the month, but if you forget about it, just subscribe on iTunes. That way it will automatically download in your iTunes. And if you're already in there, if you could rate and subscribe, that would be fantastic. So all the links I mentioned are on dnapodcast.com. Thanks for listening, and join me next episode to learn and discover new advances in the world of genetics.